Hello, friends. I am experimenting with this technology. Hopefully, I have everything set up, so we'll see how it all works out. I'm starting the next chapter, which is basically going to be amino acid structure. Obviously, the amino acids are the building blocks of proteins. Proteins are the building blocks of the cell, and the cells are the building blocks of multicellular organisms. So, proteins are where it's at, and ultimately amino acids are the center of that. So we're going to go through and talk about the amino acids. This is going to be basically a run through of the 20 amino acids and the chemistry that is particular to all of the amino acids. And then hopefully I'll stop in exactly 12 minutes so that we can move on to some of the good acid-base chemistry that's associated with the amino acids. So that's a plan. I'm trying to work with what has been described as clearly a generational, deficiency in attention span. So here is a list of several proteins. These proteins just differ by the number of amino acids and, of course, the composition of those amino acids, and therefore they have different function. Now, one of the central tenets of biology is that the structure of a compound is related to the function of those compounds, and there's no place that is more evident than in amino acid and protein biochemistry. So these are the amino acids. There's 20 that we have in nature, and they differ mainly by what's called the R group or the side chain. So all of the amino acids have one thing in common, with some exceptions, but they have an alpha carbon, so a central carbon here. They have a carboxylic acid group, COOH, and they have an amino group, right? And as I said in the last lecture, the carboxylic acid group and has an ionizable hydrogen, and the amino group has an ionizable hydrogen. So that every amino acid is a weak acid at two different pKa's, one at the carboxylic acid group and one at the amino group. Now, the R group is what allows for different chemistry to occur. And so it's within that R group that we'll see the differences in the amino acids. Sometimes some of those R groups affect, let's say, the amino group or whatever, but for the most part, the only difference you're going to see is in the R group. And all amino acids are what are called Zwitter ionic, so they are neutral. They don't have a charge associated with them, but the way they don't have a charge is that they have a negative charge on the carboxylic acid group and a positive charge on the amino group. And I should say that they're zwitterionic at physiologic pH. So this differs from an uncharged compound, right? An uncharged compound would have the same charge, but here um, you have a protonated carboxylic acid group, so you don't have a charge there. And then you have a deprotonated amino group, so you don't have a charge there, right? Same neutral charge, but coming about from different ways. And what I'll show you in the next lecture is that this compound, and this is now my eighth favorite test question, this compound, an uncharged amino acid with a protonated carboxylic acid group and a deprotonated amino group, doesn't exist in nature for the most part, at least with any frequency over 1 in 1 billion, right? And we will be able to use the Henderson-Hasselbalch equation to understand why that is the case later on. So, as I already alluded to, the structure of proteins detail its function in that we can manipulate that structure based on the sequence of amino acids. And as we know from genetics, the sequence of amino acids is manipulated by or is controlled by the nucleotide sequence in the DNA. And all of this is linked through the genetic code. I'm not going to go through this in this course because I'm presuming that you already had it in genetics. But it is through the DNA sequence that we can get mutations that cause variation that make unique differences in all of us. And all amino acids are chiral. They have at least one chiral carbon. I'm sorry, not all amino acids. Almost all amino acids are chiral. And they have at least one chiral carbon in the alpha carbon. And remember, a chiral carbon is a carbon that has four different functional groups attached to them. There is one exception to that, that is glycine. And in glycine, the R group is equal to a hydrogen. And since you already have a hydrogen in the structure of an amino acid, you no longer have a chiral carbon there. And there's some implications to having glycine. And that chirality is uh, very important. Here, this is something that we'll see later. It's secondary structure. And it turns out that having something all in the same uh, form, the L form or the D form, allows for things like secondary structure. So in this case, in this universe, everything is in the L form. And because of that, the R groups project out from this alpha helix in the same way. If we had a mixture of L and D forms within a secondary structure, you wouldn't have any type of symmetry like that. And therefore, we couldn't have these secondary structures that form, and we really couldn't have protein structure that forms. So, as I said, what we see in nature, for the most part, biologically active compounds are in the L form. We can make the D form. It's not hard to do. It's supposed to automatically draw arrows, but I guess it's not. Um, but they are not biologically active in our systems. And I'll go through in one of our live class sessions and show you how you can differentiate between the L form and the D form. But essentially, the L form, if you so this is a list of the 20 amino acids here. This is the three-letter abbreviation, which should be relatively obvious. Oops. Um, serine, SER, cysteine, CYS, and then this is the single letter abbreviation. Now, the single letter abbreviation is very useful, and that's what you'll see in protein structure, or protein sequences and things like that for the most part, because obviously it's only one character in which you can do. Now, single letter abbreviation kind of has some rules, and these are things that you just have to memorize or know. For the most part, they try and stick to the first letter of the amino acid is the single letter abbreviation. So glycine, G, alanine, A, so forth and so on. There's an issue in which there are some amino acids that share a first letter. So something like phenylalanine starts with a P. Well, so does proline. So that doesn't work. And so there they try and go by a phonetic nomenclature. So phenylalanine, the single letter abbreviation is F. Tryptophan, the single letter, because T is used by threonine, the single letter abbreviation is a W because evidently, I don't know, if you say tryptophan with the lisps, it sounds like a W. I don't quite know. For those where, for instance, the T's, where you have quite a few, then they go by the second letter. So here, tyrosine, the single letter abbreviation is a Y. Um, I don't know how they got D for aspartic acid. Honestly, that just like seems like a stretch. Here for glutamine, they went with G, but then they went to the thing closest to G, which is E. So in general, it's single letter, but there are going to be some things where you just have to kind of know. Glutamine, they say it sounds like a Q, so they go with a Q for that one. Um, Lysine here, L is used by leucine, so in lysine, they just go with K because it's close to L, that sort of stuff. This is useful, I think, for me. I will tend to use um, the three-letter abbreviation for short polypeptide sequences, but single letter for long polypeptide sequences. And these are the classifications of the amino acids in terms of their chemistry. And as I said before, the chemistry is more important than the sequence and the structure. You're gonna have to know the structure, but understanding the chemistry and which amino acids can maybe compensate from each other or would have a deleterious effect are going to be much more important in terms of protein structure and in terms of this class. So we have the charged amino acids, and those can be either positively or negatively charged. We have the hydrophilic amino acids, and remember we said that those are going to be polar. We have the hydrophobic amino acids, uh, which will be nonpolar, and then we have the special class of the aromatic amino acids. And if you remember from organic chemistry, the aromatic amino acids have some sort of ring structure associated, some sort of benzene ring-like structure that is part of the deal. So now we're just going to go through each of those, and then I'll point out, maybe some interesting things that pop up. So this is in this case, these are charged amino acids first with the negatively charged amino acids. That's aspartic acid and glutamic acid. And if you look, so we should be able to identify, any amino acids just by seeing their salient features, right? So here is the alpha carbon, right? And let me just change the color here. Here is a carboxylic acid group. Here is the amino group. And now, let's go green. This is the R group. And if you note, for aspartic acid, you have a methyl group, and then you have a carboxylic acid group. And for glutamic acid, you have two of these methyl groups and then the carboxylic acid group, right? So both of these negatively charged amino acids have a carboxylic acid group in their R structure. The positively charged amino acids, again, we should always be able to identify alpha carbon, amino group, carboxylic acid group, and then this is the R group. Boop, boop, boop. And so for the positively charged amino acids, you have lysine, arginine, and histidine. Right? So both of these will be positively charged at physiologic pH. For the polar amino acids, the hydrophilic amino acids, again, alpha carbon here, you have serine, which has a hydroxyl at the end that makes it polar, threonine, so this is the basic group, so this is the R group here, threonine, which has another hydroxyl that makes it polar, cysteine, that has a sulfahedral group that makes it polar, asparagine, our group here, and glutamine. And I'm sure, as those of you who are paying attention, you see that asparagine and glutamine look a lot like aspartic acid and glutamic acid. The only difference is that one has this amino group here, technically a guanidinidyl group at the end of it, whereas the other one has a carboxylic acid group. So those are related except for those two things. For the hydrophobic amino acids, we have glycine, which isn't really hydrophobic, but it has a hydrogen as an R group, and we need to stick it someplace. This is the smallest amino acid, and it's the only one that's non-polar. That's non-chiral. Um, yeah, that is not chiral. Alanine has a methyl group as an R group, and this too really isn't hydrophobic, but it just needed to be put someplace. Proline, which here, this is the alpha carbon here, so the R group actually comes back and makes a connection with the amino group there. So proline is interesting because of the stereochemistry that it forces onto a polypeptide chain. Valine, notice it's full of hydrocarbons, which makes it nonpolar. Leucine, again full of hydrocarbons. Isoleucine, obviously from organic chemistry, there's a minor difference between leucine and isoleucine. And then methionine. Methionine has a sulfahedral group like cysteine, but because it's buried within the chain of the R group, it is a nonpolar molecule. And we know that methionine is the amino acid that starts polypeptide chains. So it corresponds with the start codon. And then the last group are the aromatic amino acids. As I said before, they have some sort of ring structure. Phenylalanine, right, which has a methyl group, and then this ring here. Tyrosine, which is exactly the same as phenylalanine, except it has this hydroxyl here. It kind of makes it a little polar, right? It gives it some polar characteristics. And then tryptophan. Tryptophan is the largest amino acid, and it has this large ring structure in it as well, nonpolar ring structure in it as well. Okay, so I will stop there with that description of the amino acids, and now we'll look at some of the amino acids, at least in the next lecture, we'll look at some of the amino acids that have ionizable side chains and what the implications are in our favorite titration curves and Henderson-Hasselbalch equations.